All right. Welcome, everybody, to the February 2022 Hawk presentation. We're the Habitat and Wildlife Keepers. We're the first chapter of the North Carolina Wildlife Federation in Matthews, North Carolina. Started about 16 years ago and had a lot of fun teaching folks all about really loving nature. Everything from the big stuff to the little teeny tiny stuff. And we're going to talk about some pretty small things today, fungi, soil organisms. You don't see them until their mushrooms pop out of the ground. And so many people are worried about that, but we're going to have a wonderful world-renowned researcher that's going to talk about that here in just a little bit. I'm going to introduce Trad in just a couple of minutes. But in the meantime, we started the group over at Squirrel Lake Park in Matthews. This is a planting garden bed not far from the lake that you can see in the back there. And we are always happy to have folks join us and come and assist us. We're a volunteer organization. We'd love to teach folks how to go ahead and certify their yard and come on out and understand the real simple things. It's food, water, cover, place to raise young. And the fifth thing, which is going ahead and being very gentle with the chemical use. So anything with a side at the end of it, right? Herbicide, insecticide, all those kind of things. You've got to use it very sparingly, not blasting it all over the place. And being able to certify your yard, getting a sign out there, educating your neighbors on what that means would be really fantastic. So it's pretty easy. I mentioned that we're a volunteer organization. We need volunteers for all sorts of things. We have a Kids in Nature Day and Earth Day events in October and in April. We've got the leadership team that goes and organizes all these meetings, including like this one tonight. When we used to meet in person, we had, of course, refreshments and that type of thing. For now, we don't quite need a refreshment coordinator, but we do need folks to help us in all different kind of ways, as well as weeding those gardens that we have. So reach out to us, let us know how little or how much you'd like to volunteer, and we'd be glad to have you. That's no doubt. Earth Day is coming up, and we've got our annual Earth Day Festival over at Squirrel Lake Park coming in April the 23rd. So we're going to need folks to help set up. We're going to need folks to help man some booths. So please reach out to us. We're going to have a sign up for volunteers with specific duties and whatnot. So, but reach out to us if you're interested to help us out. Everyone knows about monarchs. Thank goodness their decline seems to have gotten a little boost this last year. So more people are planting milkweed and maybe spraying a little bit less in their yards. But it's not just the monarchs that is bird food. It's all of these. So all these insects are really not insects and bugs and problems. They are food for our birds and other creatures that build that food web. So be gentle with those insecticides. Doug Tallamy, if you guys aren't familiar, did a wonderful presentation for us last year. If you didn't get a chance to see his presentation, go to our website, hawkncwf.org. Our YouTube, it's on our YouTube channel. And matter of fact, all of our presentations through COVID this last almost 2 years have been recorded and put on there. So if you're not familiar with Doug, that last statement talking about the birds, the insects are bird food, it really hits home. So he's got some great books, a new one as well. If you haven't gotten it from your library, I'd really encourage you to. Our sponsors have helped us tremendously make the events that we can hold and all of the fun projects that we do. William Suber has been our Earth Day Festival keynotes sponsor for the last 6 years now. Of course, the last 2 years no, we haven't had the event, but we're looking forward to this one coming up in April. So we'd love to have you guys come, and we thank our sponsors for helping us out. So getting to tonight's presentation, Trad Cotter, if you guys don't know Trad, he is a world-renowned mycologist. He is, if you haven't heard him speak before, you're in for a real treat tonight. You're in for a real treat whether you've heard him speak or not. And you know, whether you need to learn about how to forage for mushrooms, because you know all these mushrooms are edible. Unfortunately, some of them will kill you. So some of them will make you wish you were dead, and the other ones will feed your soul and take care of you and take care of your health in a wonderful, wonderful way. So we're going to learn all about remarkable activities that we don't see under our feet in the soil, and it is at the base of every living thing here. We have no trees without mycorrhizae. So in any case, Trad is in South Carolina. He's in Jamaica. I imagine he's in a couple of other places that the Beach Boys may have sang, Bermuda, Bahamas, I'm not sure. So I look forward to hearing this, maybe even more than all the rest of you guys. We've got a nice group of folks. Like I mentioned, if you have any questions that you want answered, we're gonna have a little Q&A at the end of Trad's presentation. Trad, if you would, go ahead and turn your camera on, and you should be able to take launch your presentation and share. And I will pass it on to you when your screen gets up. And if any of you guys are in the North Carolina area, you know, actually one of the nice things about you know, these last 2 years is that our Zoom presentations have had folks from all over listening and watching and reaching out to us and learning along with us from all of our presenters. So this is, you know, I guess a silver lining, if you will. But if you're in the area, you know, and you haven't visited Trad's place down near Spartanburg, it is fantastic. What a treat to go down there and walk the trails with all these logs. And so it's Mushroom Mountain down there. So I'm sure that'll come up here in a little bit. But in any case, Trad, I'm looking forward to it. Welcome.
Can you hear me?
We can. You're a little muddled, but is your mic nearby?
Get closer to my face.
There you go. All right. Well, welcome and thanks, and I will let you take it away.
Right on. Thanks everybody for coming tonight. This is one of my passions, so this is not work for me at all tonight. I absolutely love fungi. I've been studying mushrooms for a long time. The running joke is that, you know, I've been growing mushrooms for, you know, 29 years, legally for 25. But we'll talk about that later, not back near the end. This lecture it covers a lot of cool things that mushrooms do. I titled it Mushrooms, Molds, and Mycorrhizae because they're all fungi, and there's so many other different types of fungi that are out there. So I hope this gives everybody, the participants, some insight that I have gained, not just by myself, but by like following other mushroom enthusiasts too, to give them all credit. And listen, it's not too late to forward the Eventbrite to your friends because it's going to get really interesting really fast. I'd love to see the participant numbers jump. 212, that's a great number. And thanks for coming tonight. So I'm gonna turn off my video so you don't need to see me scratching my nose. All right, there we go. We're gonna actually take a poll to see how many people were wearing pants tonight, you know, or not pajamas, but we're not gonna do that. So if we gaze deep into nature, we'll find the solutions for everything. Yeah, that's a pretty profound statement. And I like to believe and think that this is really the way that I wake up every morning. And you know, when you look at biomimicry and you look at how we can learn from nature and the patterns in nature, it's really a system that has worked for millions of years. And So the world has been through a lot, and it's been through extinctions, devastations, desertification, and all these different things. And the colonization of land was initiated by mushrooms, and they're land colonizers. They're soil creators, and we wouldn't have trees if we didn't have fungi and lichens and things like that. So they're very critical to the environment and the ecosystems. I study interkingdom interactions now, and this wasn't me 20 years ago. This is who I am now, and everything. It sounds hippy dippy, but we all know everything is connected, and the animals and the fungi, you see them in the upper right corner, are branching off from a very similar node of the eukaryota, and we share some very similar attributes to fungi, animals and fungi. Let me find my pointer. Here it is. You're right. So this is a micrograph by Patrick Hickey and Nick Reed in England. These are some very hot tips here. Mushrooms create heat, carbon dioxide, and they sweat. And these very hot tips, these Spitz and Corpus cells here, are very intelligent cells, and they can sense and detect different chemical variations in the soil or their environment, and also other biological competitors or predators. So I love this. Living on the edge here is where I like to look, and I like to think. They're essentially burning their way through the environment. Lichens compose as much as 60% coverage of all the landmass in the entire world. Not many people know that. And lichenized rock, it's algal, fungal, bacterial component. These are widely misunderstood. And there's fungal components here. There's over 5 million species of fungi estimated to be on the planet. And there's really only about 160 to 180,000 that have been identified or characterized by DNA. These are Ascomyces. These are little cup fungi and lichens, just absolutely beautiful. And so if you're ever hiking, I know there's a lot of birders here, and landscapers and things like that. And if you just get a hand lens and just go out and start looking at these microfungi that are scattered among the logs, they're absolutely beautiful, and they're decomposing matter. There's mycorrhizal fungi, these tiny little pink chanterelles. Every mushroom is producing millions of spores a day, and they're adhesive. They're tumbling around like tumbleweeds. They stick to things that they want to eat, that they want to decompose. Once that, they do spore germination, and most mushroom cultures or fungi will germinate and then they will mate. It's I hate to say this, but I'm trying to keep it PG-13, but it's like a nightclub. Some are attracted to some are not attracted, and then they're going to germinate and mate. They're going to mate and turn into mycelium, which is is a viable culture to produce another mushroom. And then they're going to colonize an area, and then they're going to produce a mushroom when they run out of space. All right. So mushrooms create heat, carbon dioxide, and they sweat, and they swim through their environment, and they're always looking to colonize, mine the resources until they're gone, and then they find a new place to live. And that's why I say, it's not hard to think like a mushroom, because they are kind of animal-like. They're very human-like in nature. Now, to identify mushrooms, I do teach ID mushroom courses in person online. So don't eat any mushrooms in your yard that you do not know what they are. This is a spore print. This is one of the processes of identifying a mushroom. This is a King Stropharia, and it produces this beautiful spore print within 24 hours left overnight. So every mushroom has a specific spore color. We can take those spores and we can just on a Petri plate that has malt or potato or whatever in the gel. And we can germinate those and we can get a pure, or we can clone the mushroom if we like, its particular habitat, or it's growing on something unusual. And sorry, my cats are destroying my place. Yeah. Commercial break. And so when we experience something like this, the environment is not sterile. So this is actually pretty profound. This is come back later when I talk about medicinal stuff. So I noticed these plates years ago, and this is an aspergillus mold, and I had a mushroom culture growing towards the aspergillus, and then it just surged over the top of the aspergillus, which was just phenomenal to watch. It was like watching, I mean, I don't watch football, but I guess for some people it'll be like watching the Super Bowl. And this is what I would do on Super Bowl Sunday. We'd be watching my Patriot plates. And this would be the halftime show. And then it was perfectly symmetrical. And then it got out of its symmetry and surged over the top of this aspergillus. You can actually see a slight depression there where the mycelium is pushing down on the mold. And then you see these little droplets down here, which was what I noticed when I zoomed in on the photos. And that typically is not there in pure culture. So what I did was I developed a method for trying to investigate what is going on here. And there's the plate, like, a day or two later. It completely surged over the top of this aspergillus, another biological competitor. This is what's going on in the soil. This is what's going on in your garden. Mushrooms are, most mushrooms are protectors. There are some parasites. And they're just basically carpet bombing this other fungus. So then what I did was, this is probably on a Saturday night, big night for trad, is I cultured out, this is elm oyster, no, this is shiitake, and this is elm oyster. And then I pitted them against each other and made a little gladiator match. And I noticed these beautiful droplets of metabolites forming on this little DMZ here. And these droplets are filled with antifungal compounds that were dialed up to specifically kill the shiitake fungus, which I thought was just fascinating. So mushrooms produce sweat. In that sweat are chemical metabolites that have enzymes. Mushrooms feed on the outside of their cell walls, and they feed by absorptive nutrition. So what they do is they sweat. It's almost like your stomach turned inside out. They'll sweat. It'll break down the environment or the molecules, and then it will pull right through its cell walls what it needs to survive. All right, so it's absolutely fascinating. A lot of the native ground orchids, actually, it's almost undebatable at this point that all native orchids in North America have some kind of fungal association with the seeds not being able to open by themselves. And so what happens is there's a tiny little fungus called Rhizoctonia, which is also associated with the decline of, it's a root rot for a pine tree. So whenever you see these beautiful native moccasin orchids, you will also see a dead pine tree nearby because that has to have that Rhizoctonia-like organism nearby. Just kind of prise them open like an oyster knife, which is really cool. Mycorrhizal fungi. This is mycorrhizal, so fungus and root fungus. There's thousands of species of mycorrhizal fungi, and I'll give you a few examples. But these are fungi that thread into the root system of the plant itself. And this is a very ancient association that has probably lasted over about 400 million years that they've found in the fossil record. And it's a symbiotic relationship where the fungus threads in, increases the surface area of the root system, also provides pathogen resistance and nutrient absorption. 30% more nitrogen, as much as 60% more phosphate. And, we are approaching peak phosphate, meaning all the phosphate in the rock and that we're using for agriculture, we are going to run out. It's not if, it is when, and they've already predicted within 30 to 40 years, it is gone. So fungus do that naturally. So this is something that we have to think about, is that if you are a gardener, if you enjoy planting, if you're a landscaper, we've got to think about partnering with fungi in our gardens and our landscapes, because there will be no phosphate, right, on the planet. This is my axle of evil number one. You know, this is the natural fertilizers from natural gas sources. This has to end. And when we fertilize our gardens, when we fertilize fields, they're essentially, you know, it's fast food for plants. And if you fertilize with high nitrogen, especially phosphate, over a certain part per million, then the fungi, the mycorrhizal fungi, do not associate with the plant. The plant doesn't even need it. It's like, I don't need you. So what do? They just take the phosphate from the fertilizer and they don't need it from the fungi. So they don't allow the fungi into the cells at all, which is a really bad thing. So we have this kind of depletion and dependency on synthetic fertilizers and phosphates. And so the soil microbiology is essentially really disrupted. And this is essentially dead soil, is what I would call it. We've got so many beautiful mycorrhizal fungi. This is the East Coast. These are black trumpet mushrooms. These are some of the edibles. Fruit in the summer. And I love going mushroom hunting. I just don't go enough. You know, this is where I'm the happiest, is in the woods. And I'm out looking for fungi and bonding with them and learning about what to do based on what I see in the woods. And it really translates really well over to my research. These are chanterelles. They're really, really an amazing edible mushroom. But they do so many good things for the trees. You know, not only are they good food for us and squirrels and maybe an occasional deer, but they are essentially mycorrhizal, mostly on oak trees on the East Coast, conifers out Midwest to West, and lobster mushrooms, which is a very bizarre composite organism. This is a russula, very large, white, inedible russula mushroom, and it's covered with a mold. This is a hypomyces. It's a reddish mold, and it transforms the mushroom into a beautiful, like orange, you know, up like structure in the woods, and it tastes like lobster or seafood. It's just crazy out there, and that's why I love going out there. It's like deep sea fishing. I don't know what I'm going to find, don't know what I'm going to get, and it's like a scavenger hunt. Morel mushrooms, completely hollow up to top to bottom on the inside. This is a cup fungus, so it's all a series of cups. This is probably one of the most recognizable mushrooms in the world, especially culinary, and very hard mushroom to cultivate, but it's not so hard to find if you know where to hunt. This is a very large morel. A firefighter called me and said he found 60 around one dead elm tree. And so associates with dying or collapsing tree colonies like elms and ash trees. And some of you may know or may not know that the ash trees are on decline because of the emerald ash borer, which I'll talk about later. This mushroom is benefiting from another tree's demise. Micromediation is a big word. It's essentially bioremediation, which is the breaking down of environmental pollutants using fungi, right? But it's not just fungi that do this. This is also bacteria that work in concert to orchestrate the remediation process. So it's like an epoxy set. You need fungi, you need bacteria. There's natural attenuation that occurs. This is problematic because man-made pollutants are always hitting the soil. Something like this, a company that was paid for bioremediation would come in here and they would scoop this up and then they would discard it or it would be incinerated. So it wasn't really remediated at all. It was just, there's removing toxins and there's moving toxins. So typically toxins and pollutants are just moved. They're not removed. I've been on the phone and on some phone calls. I don't know if anybody's been watching the news, but there's been a huge oil spill in Peru after the tsunami from the Tonga volcano disrupted something in Peru. So we've got a task force together to try to look at something like this. Those enzymes, when mushrooms run out of food, they get desperate, and then their enzymes, enzymatic systems just start sweating and start looking for material. It's a molecule. It's not a stomach. They're just looking for stuff to break down and bring right through their cell walls. And hydrocarbons are just fine. Pesticides, hydrocarbons, bacteria, E. coli. This is South Carolina. So E. coli is 50% protein by weight. It's a huge nitrogen source for something like a mushroom. And so they have the ability to stun and kill E. coli and use them as a nitrogen source. These are the fecal coliform impairment watersheds in South Carolina. This is the human populations in blue. But I will also tell you that if I was to put up the slide, I don't have it anymore. I don't know where it went. But the cattle population fits perfectly in this purple area. So agriculture is a big problem. I used to have chickens on a farm, and a lot of people have chickens, but there's some farms that have thousands and thousands of chickens. And we can inoculate their floor with mushrooms. I learned over at Clemson University that in Vibrio cholera, this is just an example that we can actually filter the water if we put mushrooms in place. Imagine a garden or in your little, you talked about a rain garden, you know, to attract birds and different wildlife, that if you have fungi in there, whether they show up naturally or if you invite them or plant them with intention, you will have a micron filter set up there that can filter water before it leaves your property and goes off on the next piece of property and then into the watershed. So this is a Vibrio cholera. This cholera is a pathogen. This one actually sticks to the mycelium. Now, mushroom mycelium, remember that's the cell walls of the fungus, is composed of chitin. Chitin is equivalent to the molecular structure of crabs and diatoms, copepods, you know, all these different things. And this bacteria produces an adhesive, like a glue, that the bacteria sticks to the fungus. So not only is it a physical filter, like a micron filter, where it physically traps it because it's too big, you know, the bacteria is too big, it'll actually collect it and it will glue to the fungus like a magnet. It's just beautiful. We did atrazine studies several years ago. Atrazine, I did my fellowship with the EPA, and we looked at some different compounds, including atrazine has been red flagged for the United States. It's getting into the groundwater and doing a lot of damage. The genetically modified crops in the United States are being bred and engineered to be resistant to atrazine. So what's happening is that airplanes, sprayers are spraying atrazine at records amounts on cornfields, soybean fields, and that is going into the ground and into the groundwater, and then the residents are getting sick. You know, they're getting exposure risk from atrazine. So what I did was I did the labeled rate, I did double the labeled rate, and then I did triple the labeled rate. And then an oyster mushroom culture, by the way, and it ate up the labeled, doubled the rate, but at the tripled rate, it just sat there. And I'm just looking and watching it for 12 days. And it just sat there, and it's kind of cool that it grew out in the shape of a mushroom, but that was insignificant, but kind of cool. It sat there for 12 days. That fungus should be out to this here in 3 days, but it just sat there. put it in the incubator, came back 2 to 3 days later, and then boom. So now we're looking at this fungus. Fungi potentially have the ability to evaluate their environment and dial up a cocktail of enzymes or antibiotics, whatever they need to survive, because they need to harvest atoms and molecules from their environment. They're able to process pesticides. That's great news. And also on the recycling front, I just threw this in. I threw a couple wild stuff in here. Recycling, you can actually grow mushrooms on different things. The reason I did this was because 30 to 40% of household waste is suitable for mushroom composting and cultivation. 60 to 80% of a business or a school is suitable for mushroom recycling and cultivation. That's a big number. And that is tonnage of material a year that is not going to make it to the landfill. There's no reason these things have to go to the landfill. They can be composted right on. Hey, and you get some edible mushrooms as a byproduct. That's a double win. You know, a lot of material landfill, you can. This is an easy experiment. You can start with coffee grounds, order a oyster mushroom culture, and just start in a bucket and just add the coffee grounds. Sure, you can compost this. You could put it in your garden, but you know the mushrooms colonize this, and they enhance the microbial attractiveness to this, especially with worms. And so we're going to fruit mushrooms like this. This is mushrooms growing off of coffee grounds. You can see the natural fibers here, the natural filters, unbleached. And then you got mushrooms popping out the top of this little pickle jar in just a few weeks. You can eat those. There's as much protein, calorie for calorie in oyster mushrooms that there is in chicken or eggs. It's a great protein source. The only non-animal source of vitamin D. You're looking at it. I just did this the other day with Heidi, my little girl. She's 8. I mean, we're out picking out eggs, and there's eggs that are packed in styrofoam containers or plastic. And I'm like, we opted for cardboard ones because we know that we could pack this with coffee grounds or shredded paper and then add a mushroom culture to it. And we can get fresh mushrooms growing off of our home waste. And then we can add it to the compost pile and feed the worms later. I like this one. Cereal boxes. I did this one. I walked into a mellow mushroom pizzeria. Why not, right? This is irony on top of irony with irony in between. And I asked the owner, I said, can I have a couple pizza boxes? Because pizza boxes are not recyclable. They're covered with grease and cheese. Well, guess who else likes to eat mushrooms, cheese and grease and pizza? Mushrooms. So we filled these with coffee grounds and paper, and we added the culture, and these beautiful blue oyster mushrooms popped out of the boxes. And these 3 boxes actually fused together. They were stacked, so they fused together. And then we just started to explore like different ways to grow mushrooms on different waste. And this is out of my book. We just wetted some cardboard, and we cut the end of an oyster mushroom off from a store. And I've done this in Haiti, and I've done it in Jamaica. I've just find some store-bought oyster mushrooms or somebody that have grown them. You can take the base of a oyster mushroom, wrap it in wet cardboard, and look at this. You can get a starter culture. And this little burrito that I wrapped up, you can tear this up and add it to your coffee grounds. And then that 1 bucket of coffee grounds can make 10 buckets of coffee grounds. So to me, it's almost infinite. You know, it's just infinite. This is a bucket full of shavings from a cabinet maker, you know, that was my grandfather's neighbor who had a pile like 12 feet tall. I'm like, oh my God, we could totally grow mushrooms on that. You know, we just have to know what to do with it. Growing mushrooms is not that hard. This is me a little bit younger. God darn it, I have no gray hair in that photo. But anyway, this is golden oyster mushrooms. This was a study that I did growing off of peanut hulls after the earthquake in Haiti. It only took about 2 to 3 weeks to produce those mushrooms. Growing on wood chips and compost, this is important. This is probably one of the most important sections of the talk because we have all this debris, we've got land clearing, and King Stropharia is by workhorse in this arena where this mushroom is native to the Southeast in the United States, and it makes soil very efficiently. It fruits in the spring and the fall. It likes hardwood wood chips. So if you have the ability to get some hardwood wood chips that are fairly fresh, meaning within a month, you can wet these chips, inoculate them, put them around your edible garden, your edible landscape, and then you could add some spawn to them. And then you can make, these are intentional beds out in the woods. These mushrooms can get fairly large. They'll get, for every inch of mulch depth, you will get an inch in cap diameter, which is pretty dynamic. So they'll get up to 2 feet across, but 6-inch, mulch depth, 6-inch cap, good grilling size. They'll pop up all over the place. It's native. You also have a bluet, which is a very silky, sweet native to the Southeast. And this one likes more lignin-rich, like composting bark and leaves. And I always get totally annoyed with people. Sorry if anyone does, but maybe I'm not sorry, that you put your leaves out on the curb, it just goes away somewhere, maybe to a composting facility, maybe a landfill. And that happens. But I mean, there's a reason they're called leaves. You need to leave them there. You leave them on your property, break them into beds at least. But the trees need those leaves. These bluets, these native mushrooms need those leaves to break down and to turn into soil so the trees have some nutrients, right, for the next progeny of acorns and seeds to germinate in. Another good mushroom called the almond portobello. This one grows in your garden. This one is native to the United States and the Southeast. This was the mushroom that was grown by the early colonists of the United States in stable manure. It was a heat-tolerant native mushroom related to the white button and the portobello, same genus, and it's a very delicious edible and potently medicinal. There's some cancer compounds in this one. Mushrooms produce heat, carbon dioxide, and they sweat. All right. What also comes out as a gas would be octanol. This is a mushroom alcohol that kind of hisses off and evaporates out of the tissue and the mycelium. This is important. All right. This is important. This is chemically identical to what comes out of human skin, out of a sweat gland of a human or even animals. This is how mosquitoes find you, right? Octanol is a chemoattractant for a lot of different organisms. But what's interesting is that I attended the International Worm Symposium, and that was a lot of fun. But someone there said worms are attracted to octanol or fungi. And so I figured out why. And then we started composting our mushroom mycelium with worms. So this is what happens in your garden. If you have mushrooms, you should be welcoming fungi in your garden. If you don't have mushrooms in your garden, if you add wood chips, add some mushroom spawn. These worms can smell mushrooms, mushroom mycelium from like 10 or 12 feet away, and they will vector their way to you. Why is this important? Soil creation is a huge problem. I'll give you an example for the upstate South Carolina. It's easy. Upstate Greenville, South Carolina, the topsoil depth was 12 to 15 feet deep in 1910. The Trees of Greenville did a study, nonprofit group. They went back and checked in the same spots. It went from 12 to 15 feet to 3 to 5 inches. That's gone. It is gone. It can take 500, at least 500 years. They say sometimes 800 to produce 1 inch of soil in a healthy forest ecosystem. So if you do the math, it would take 70 9,000 years to put back the topsoil that we lost in 100 years. I mean, it's a little bit, I don't want to say disturbing, but it's an alarming statistic. Mushrooms can create soil very fast, very efficiently within a year, if you know how to do it. So we have these habitat loss. The city of Greenville, fastest growing city in the United States, has lost 75% of all of its tree canopy in the last 40 years. The city of Greenville, that's the city in my backyard. But this is what I want to see. This is what I want to see is habitat renewal. I want to see soil creation. Without soil, we don't have trees. We don't have trees. We don't have, wildlife. We don't have insects. They're going away. It's a domino effect. So then we start looking at what's called mycopesticides. These are awesome. I like these. So these exist as a mold, but they can also jump the track. So I'm going to use my laser pointer here to show you the figure 8. You all see the figure 8. I'm going to go around the figure 8. So they exist as a mold. They'll stay down here on the green for a minute, and they're a mold. Well, along comes a spider, or an ant in this case. It sticks to the cuticle. It recognizes the cuticle. So these are fungi that actually stick, and they have to chemically identify with the chemical structure of an insect's armor, and then it penetrates it, relocates the insect, and then mushrooms pop out of its brain and all over its body. And these are very target specific. So I'm gonna get out of this slide. So let me just show you the drama here. This is an ant. This is in our backyard, absolutely beautiful. And you can see this ant climbs up the tree. The fungus infects the nerves and the muscle tissue of this ant and gets into the brain, threads in chemical signals to tell this ant to climb up a branch, bite into the bud scar. You all see that little curved area, bites into the bud scar. The fungus melts the jaw muscles of the ant, and then so it won't let go. It kills the ant and then threads into the tissue of the tree, and then out of the back of its brain pops a mushroom directly over the trail that the rest of his buddies are using. It's like dropping a dirty bomb right off of I-85, I-95, I-26, whatever interstate you're using. It's pretty incredible. It's absolutely incredible. These are aphids that are, again, you see them kind of growing along the venation, biting into the venation of the underside of the leaf. So we find different ants. This was photographed by Todd Elliott up in North Carolina, just absolutely gorgeous. They call them zombie ants. These are in your backyard. Go out and look. Look in your backyard. Look under the branches of your trees. They like hardwood trees. Typically, I found them on sweet gum, beech trees, and also American holly, mostly, right? Not on the leaf like this, but under the branch, like you saw. There's spiders. These beautiful spiders that are just absolutely mummified. And, you know, we love spiders, beneficial. But there are fungi that are doing this to our insects here. They're keeping them in check. The insect kingdom is the largest one on the planet. All right, fungi are number 2. They're battling it out here. This is a cicada nymph that I found. That's the only photograph ever been taken. And we've seen different products on the market. There's one called MET52. So if you're a beekeeper or a wildlife, this is actually very important. There's a fungus called MET52. A big company. I don't want to say the names on here. A big company that you probably know by name starts with MON, and then they bought the culture from a company called Syngenta, and now they own it. But this one was targeting varroa mites on the back of bees, and now it's been completely, almost entirely removed from the market, which is unfortunate because that is a biological control for something that is targeting a parasite of bees, and the bees are the vermites are also injecting viruses and things like that. So look up MET52, capital M, MET52, and just see what's going on with that. We found some scale insects. These are on twigs in the backyard, on sweet gum, and then you see these little antlers coming out. So if you spot some insects, they're bizarre. Again, you're probably looking at these like, they're not in my yard. Yes, they are. They're just so tiny. You just really need to take the time to creep along and look at some of these things that are going on. This is a cordyceps mushroom growing out of a carpenter ant. It's huge. It looks like a hornet. This was my first cordyceps that I ever found. It was phenomenal. I couldn't believe that I found it. 2 mushrooms growing out of her right above her thorax, which is like right above your neck. And this is those that broke off, and then she's fully mummified. So again, if you see an insect that's mummified, white, or has structures growing out of it, photograph it, send it to me, and we can take a look and see if there's any particular, environmental or biological use for something like this, because they are not easy to spot. We took this one, we cloned it, we cultured it. Again, it turned back into a mold and turned into these beautiful clouds. And this is exciting because, this is just insane. What happened next? Again, my first cordyceps, Camponotus, which is carpenter ant. I looked up the phylogeny of the carpenter ants, and they're very closely related to the clade of Solenopsis, which is fire ants. And so we reinfected Camponotus, some more black carpenter ants, and it was the pathogen. It infected them within 4 to 5 days. Then I started looking at the clades here, and I noticed that Solenopsis and Camponotus, they were all in these clades down here, very closely related. But then I started looking at these insecticides that are on the market with the EPA. This is the EPA. This graph was taken directly off the EPA site. All right, acephate is orthene. All right, that's what's sprinkled on ant mounds. There's a lot worse than that, which is this one. And 7.5 billion pounds of this product is used per year. That's the active ingredient of that compound. So when you look at an ant killer, it could say 0.5 or 1.2%. It's usually not that high. So just think about how many pounds of this stuff is put on the US soil per year and what's going into your drinking water or well water. Fire ants are on the move. We've got some mild winters, and we've got a few ideas here. So what we did was I made, one year I made fire ant farms in the house. I don't know if everybody has fire ants. We we sprinkled the culture onto the top. This is post-mortem here, but then the ants tunneled around for a minute, and then that Camponotus, meaning the carpenter ant culture, was active against fire ant culture. That's a big deal. And it happened within 4 to 5 days. And we did a couple field trials at our house. We did some at the farm, and we're still fooling around with the formulations for this. But just think about this. We found a native fungus growing on an infected carpenter ant and reintroduced it to a fire ant within this state, and it mummified them. An invasive species. This is how a fire ant should look. And if you agree, say hell yeah in the chat box. So that brings us into other things. It's like, listen, okay, but that was a game changer. If I saw or found a carpenter ant and it mummified invasive fire ants, what about this one? Does anybody know what this is? This is a spotted lanternfly, which is in North Carolina now. It's moved all the way down from Pennsylvania. This is the #1 threat to agriculture in the United States. That's what the USDA has said. It's not good, guys and girls. It started in Burke County, Pennsylvania, ground zero. And I don't have a new graph. Sorry, I threw these the last minute here. I'm procrastinator, I'm an incubator. Pretty bad excuse. But look at this. They completely swarm trees. They came over on the tree of heaven, and they're, sap suckers. And they'll decimate orchards and vineyards. So I gave some lectures up in North Pennsylvania, and this woman was so kind, and she came to one of my lectures and she said, I spotted something that looked like a mummified insect, and that's why I give these lectures. I want everyone watching to be on the lookout for a problematic insect. I don't care if it's a cockroach or a weevil or a vine borer on squash, Japanese beetle, emerald ash borer, longhorn beetles, things that are just really messing up the ecosystems. She found 100 of these in one spot, up at ground zero. So she brought these to me. I was able to clone it, and we're able to culture it. So we're hoping to reintroduce that back up into Burke County this year, as soon as they start to reemerge from their egg cases. We've got fungus, plant fungus and viruses. You've got endophytes. Oh, sorry. There are heat tolerant grasses that grow along these geothermal springs. And they are right at the edge. It's about 150 to 160 degrees Fahrenheit. So how are these grasses surviving? This is awesome. This is bizarre. They found an endophyte, which is a fungus that's threading into the roots and in between the cells of these plants. It was Curvularia, and they isolated the fungus, purified it, and they put it back in the plant. And even when it was colonized and they heated it up, it died. But then a virologist was in the room, and they said, there's got to be something else missing when you purified it. They found a virus living inside of the fungus that was living inside of the plant. So you've got a 3-tier level of biology that was making a plant that would not survive above 100 degrees, 110 degrees, giving it heat tolerance of 150 to 160 degrees, a virus living inside of the fungus. Just mind-bending. And so you've got, every time now, if you're watching, when you look at your leaves of your plants and in your garden, you're not just looking at the plant. There are fungi threaded in and throughout the leaves. You see beautiful, bright leaves, or you're planting strains of tomatoes that have passed down the fungus from generation to generation. How do they get there? They get there through the seeds, through seed collection. So what I'm telling you is that the fungus grows through the plant, and the plant brings the fungus forward to the next generation. Where does it deposit? In the seed. It packs the fungus for the trip, for the next generation that does really well. So, saving seed from healthy plants, great idea. Well, it's because it's a composite organism. We go right back to the beginning. I also study interactions between fungi and bacteria. I showed you fungal interaction. These are some of my behavior galleries. I think these are fun. This is a shiitake fungus. These are 2 E. coli pellets. For my birthday one year, I ordered Salmonella, E. coli, strep, pneumonial cultures, Pseudomonas, Candida, and staph. That was my birthday present. So that's what I do on Friday nights. I do these little gladiator matches. I set them up, and then this is just a day or two later, the bacteria just rush over to the edge of the plate as far as they can. And these are inhibition galleries. All it's doing is kind of giving me some insight as to whether or not the fungus is diffusing some kind of antimicrobial substance into the agar, the gel that would inhibit the bacteria. All right. This is a staph. I isolated this off my body. We're covered with staph. I found some mushrooms through some research. I was looking into somebody who's studying antimicrobial mushrooms. These are false tinder conchs. This is a really cool fungus. It grows on birch at higher elevations in North Carolina and the Southeast. Not edible. You can start some fires with it, you know. So I grew this on a sawdust block, and then I started to play around with the bacteria. So I was taking throat cultures and staph cultures, dripping them into the block, and then it sweated out a novel metabolite in about 24 hours. These are the controls. I did 3 to 5 reps per session of different pathogens. But in that bag, it produced 20 milliliters of fluid. All right. Don't log off. I see the numbers dropping. It's like the stock market. It's going to get really cool soon in a minute, because we screened all these different bacteria on these different fungi. And then we test it with plant pathogens, meaning if you see a plant pathogen or a root rot organism in your garden or on a farm or a huge farm, we can take that fungus or that bacterial blight and inject it into these bags. So essentially what I did is created was these pathogen bags where you could take it from a human, your dog, your goat, your corn, cotton plant, it didn't matter. And you would inject it into the pathogen reservoir. And then what would happen is 24 hours later, you get this beautiful fluid. And what's interesting is that only 20 milliliters of this was active 10 to the 6. So if I just get 20 milliliters of this out, then that would be active if I was to make a fluid that was 528 gallons, which is enough to treat a field, right, or a small field. So we tested it on shiitake and staph, reishi mushrooms, and what is that? Strep throat cultures, turkey tail mushrooms, and salmonella, and then we just diluted them out. Active. All right, I'm an hour in, and with psilocybin. I was told I could talk about this. Also do psychedelic research in Jamaica, and I just want to touch on this because I now have permits there with the government and my attorneys to study antibiotics produced from a class of fungi that are Schedule 1. They are chemically identical to a serotonin inhibitor. You know, with the mushrooms, they bind to certain receptors in your brain. There's many types of mushrooms that produce psilocybin on the planet. There's hundreds of varieties. And so I'm based out of here, down in Jamaica, in Montego Bay, and soon moving down to Costa Rica to set up a lab and do some sessions there. I met with the Minister of Agriculture. What these fungi do is they are turning off or switching off the default mode network. To put that in layman's terms, imagine the firewall on your computer. You have built up a defense system to protect yourself, like an ego, and you can temporarily turn this off and access all kinds of very deeply ingrained information. So this is big. The #1 class of drugs sold in the United States and consumed is antidepressants. This is a tragedy, actually. It's making billions for some companies, but it doesn't fix the problem. So I don't want to get into it too much, but I would tell you that you can look up on YouTube, you can watch some studies by Johns Hopkins University, specifically Roland Griffiths. All right, and we'll get to this in the Q&A or the chat later. So these are therapeutic doses done with certified therapists and sitters. And you know, if you don't know what you're doing, this is a destroying angel. It's the deadliest mushroom on the planet, the one on the left. And this is a psilocybin. This is a hallucinogen on the right. And look at that. There's no risk of cross-contamination here, even if they're touching. But my running joke is, listen, no matter which one you eat, you're guaranteed to see a light. But you better know what you're doing out there. Mushrooms, these psilocybin down here, this is from one of my labs down on the east side of Jamaica. We're growing them fairly large numbers for a Canadian company. And of course, I've got my little Heidi here and the blue portal, and we go out looking for mushrooms. All right, native habitats, native strains on the island. There's nothing more than I like than this is a form of mycorremediation. We talked about that, about healing the planet, healing the soil, filtering the bacteria. But what about us? We need the human race to be healthy in order to do this. In other words, to fall back in love with the planet and to be able to understand the connections that it has. And it's going to come back to us. And that's what I love, is this unconditional love that you see, this bonding and connection with the universe is just a gorgeous thing. I'm glad to be a part of it. I didn't see this coming at 49 years old. I never thought it would happen in my lifetime. And here we are. It's progressing fairly quickly. You've got fungi and endophyte. Okay, remember those? Those are the fungi that grow in the leaves in the plants. They found one. I can't remember the name of the university, but they found one that degrades pure polyurethane in culture. All right, polyurethane is the backbone of plastics. So you have fungi that may have the potential to break down plastics. We have to protect our forests. We have to look at this as a Rosetta Stone. We have to pay attention to fungi. They're showing us the language. We just don't, they can't speak. We can't speak their language, but we can understand, we can observe, and we can watch what is going on because we're seeing this happen. The earth has lost half of all wild animals in the last how many years? I'm going to turn on the chat box to see if anybody wants to guess. Huh? The earth has lost half of all our animals in the last how many years? Somebody got it. Should take a poll. Click, 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 click. You're not going to like it. All right, I'm going to go ahead and hit it. You're not going to like it. "40 years. And that's what we can see. That's what we can monitor. You understand? I'll tell you one more thing. We are, scientists have predicted that we will lose 40% of all insect species in the next 25 years. The largest kingdom on the planet in 25 years? That is from habitat loss and pesticides. Fact. All right. So there's solutions there to these two problems. Habitat loss, we can solve that. Pesticides, we can solve that. We just need to replace them with something that's more biologically efficient. I'm going to stop here right now, and before I go off on a tirade. So I'm going to stop share and bring my host back. Are you able to come back, or do I need to re-add?
Nope, I can get back in.
Thank everybody for coming tonight to listen to me rant about fungi. I'm going to put it back on.
And you're getting plenty of folks that are delighted to, a lot of wows, a lot of way to goes, wonderful stuff. Ranting or not, it's exactly, unfortunately, the truth. That is some of the sad parts. There's no doubt. The fact that we're researching this and finding the answers, I really appreciate you giving a a call of action earlier. I'm not sure if everybody heard the call of action, so I'm going to repeat it. Trad was talking about getting out there and being a citizen scientist and seeing what's going on in your very own backyard or in parks or out in nature, particularly when you see the types of things that he's doing with these mummified insects. Trad, how would someone be able to get that? Is that something that, in terms of to get it to you, I mean, is that something that a regular envelope and just mail it to you in the mail, or how would they do that?
Probably not an envelope. You know what? It was really interesting is I need the insect intact, so an envelope it might get crushed. A lot of people use like little Altoid tins and things like that. Like little metal tins, something to wrap it in. I prefer you not send it to me in your prescription drug vial, which is actually a really good container. But if you do, just peel off, whatever was on it. Tic Tac container, anything like that. You wrap it very gently and maybe a little paper towel, and then just so it's not moving around and something hard, but picture first so I can see what it is, if it would be. But again, if you send something to me, I'm going to culture it. I'm going to clone it, probably. And if we use it for something, I'm hoping that I always told this to everybody, including the gal that drove down the culture with the spotted lanternfly. I'm an honest person. I'm an honest business person too. That I signed something to where if we did do something like that, and there was any money made, from that I would, we would work out a commission on something like that. And I always think of money last. I'm not putting myself out like that, but I want solutions first. I want solutions.
Absolutely. Well, good. Hey, that's even better. Not just being a citizen scientist, but maybe even getting your name on a new strain and a possible solution. We've got a lot of questions, folks. Let me ask you to put it in the Q&A. We'll try and get through as many of these before we've got to go. Again, the whole notion of what you can do in your own backyard. Is there a particular start that you would recommend? For instance, we all know shiitake mushrooms and oyster mushrooms and that kind of thing. Is there a particular mushroom to grow that is an easy one for someone starting out?
Yeah, the one that I was pressing the most was King Stropharia. Now, shiitake are very reliable on logs and things like that, but the King Stropharia is a mulch, growing mushroom, and it just does so many wonderful things for the garden, and it fruits in the spring and the fall. Yeah, you have to add more wood chips and you add more spawn every year, but you plant more tomatoes every year. It's like you just add it, and what I like about it is it builds soil very quickly, so you can keep fresh wood chips to the same area and respawning it. That seems to be one of my garden favorites. And you can grow oyster mushrooms, you can grow a lot of others, but shiitakes on logs is the easy button. And King Stropharia on wood chips is number 2, buddy. That is good. And it has so many good environmental implications to it. Right.
That's awesome. Is there
I'll
a particular...
I'll put the name in the chat box, all right?
All right. Is there a particular thing that someone can do in their own backyards? We were talking earlier before the presentation started, we kind of do a little behind the scenes, just conversation of things that we're seeing out there. And, having water for wildlife, having shelter and cover for wildlife. But the notion of a rain garden and the different, you were talking earlier about the inoculating your watershed on your property to arguably clean the water as it's leaving your property. So if it's going into a stream, if it's going into the next door neighbor's property or adjacent or whatnot, it's already starting to clean up, right?
Yeah, and it's going to, remember, biological contaminants are going to get filtered out very quickly because it's a physical trap. It's a micron filter. Things like pesticides and herbicides, hydrocarbons, those need contact time, because the enzymes need time to combust those molecules. They use those molecules, they use oxygen and different enzymes to just break them apart. So holding areas like rain gardens and little swales are perfect for that. So adding fungi on the edges. We just installed a huge microremediation and water runoff encatchment at Mushroom Mountain. And right now we are documenting everything. And I haven't seen anything done like this before. And a contractor came to us and we solved problems, but we totally used the whole idea of using fungus so we could use it as a teaching platform. In other words, we're going to document it, and this is going to be something that we can talk about, that we can have workshops there to look at the whole system. But there's a series of attachments that hold water, and the fungi are there, so it gives it contact time. So for molecular pollutants, contact time. Biological, not usually a problem. That's fairly easy. That's just a micron filter. It traps it out. 100 to 1, it's gone in a few minutes.
Is there a particular mycelium that you guys are using in that rain garden or in the swales?
Yeah, I'll put that one in the chat box. It's called Tiger Sawgill. It's a native fungus that my mom found, and it like plasticizes the mulch. It's like living glue. It behaves like an oyster mushroom. It grows really fast, and it can attach to hardwood, conifers, and it grows really fast. It holds everything together, which is important because you don't want a rain event to blow the garden out, right, if you have mulch in it. So it really holds all the wood chips together and prevents erosion, and it traps bacteria. And it's also very good with chemical pollutants. And we tested this on atrazine, which I talked about tonight, at Furman University for the past 2 years. I know that paper is waiting to be picked up by a journal to be published. So
nice. We're looking all forward to that. That's for sure. It is remarkable. A lot of folks have asked if there's going to be a recording of this, and providing that technologically we did everything right and pressed all the buttons right, and the internet's working right, then it'll be available on our website along with all of last couple of years presentations, hawkncwf.org, here in another day or two.
Hey, Sheila asked a question. She pulls up mushrooms because they look ugly. She needs to leave them. Well, stinkhorns sometimes I would agree. Just maybe not so sure. Yeah, you can leave them because you've got the mushrooms are there. They're breaking down the soil. If you pick them, you're not going to disrupt their body. Their body is you don't see it. You're not removing the fungus. I mean, you're removing the reproductive structure. But then they decompose. They become food for little, snails and crustaceans and part of the food web. They're perfectly good.
Yeah, there's no doubt. You had talked about the Met 25, Met 52.
Yeah.
The beaveromite. Is that a treatment or is that the issue?
That's a treatment. That is a strain of a fungus that was found over in I don't even know, Romania. And it's highly active against varroa mites and brown spotted ticks, which I think is very fascinating. And I have the culture. I can't sell it, but I'm thinking about dusting my dogs and cats with the fungus because it's like a natural. It mummifies brown spotted ticks. I'm not a beekeeper. But for somebody who has bees and they have a varroa mite problem and they don't want to increase pesticidal pressure on the colony because they have enough issues. I would try to find it. It's a little bit frustrating that that happened. And I don't know all the truth. I'm not going to go into a conspiracy, but you're dealing with a company who has been known to genetically modify things and put a little bit of a pressure on other products. So.
That's the most gentle way I've ever heard that said. There's no question about that.
I practiced that.
Yeah, well done. You and your attorney have practiced that well.
Yeah.
Hey, so there was a question about bees that have been seen sucking mycelium. You know of a way that the mushrooms could be grown that will give them access to the mycelium or the exudates that what mycelium they're talking about?
Yeah, that's some work Paul's done, and I've known Paul a long time. They've looked at certain polypores, and I would suspect a lot of different mushrooms do this for bees because we saw this at our farm, and there was no polypore. It was just we were soaking sawdust in a bin, and we had a little spigot on the bin, and it was dripping, so it wasn't airtight or watertight. And all the bees were just massed on this little tiny drip area that were coming out of the wood that was being soaked. So there was going on in the wood. It could have been mycelium. It could have been some yeast. But I know Paul was using some different polypores, which are conchs, the big hard mushrooms that grow on trees. And they take the mycelium and make an extract out of it. And then they feed that to the bees. So it's based on the mycelium itself. The metabolites, remember the heat, carbon dioxide, and sweat. It's the sweat that the mushrooms are producing that the bees are attracted to, right? And there's antiviral compounds in there. So if the bees are attracted to it, if they're needing it and drinking it, and empowering their immune systems, that's an incredible thing. So this is something that has probably been going on for millions of years that Paul noticed, and now it's just opened up a beautiful can of worms because now we can look into this much deeper, right? Indeed.
You talked earlier about the tiger sawgill helping glue things together, and the idea of erosion control. We've all, as gardeners, we've known that compost just in general helps glue between all the protozoa and bacteria and fungus that are in compost. It glues your soil particles together, right?
Yeah.
So is it tiger sawgill that it particularly has more of a feature of that, or is that just something that all mycelium assist in?
No, that's a good question. So not all tensile strength is identical. You've got like morel mycelium is very weak. It's the weakest one, like morels. It's like fine cotton candy. It just melts and goes away. And then you've got, shiitake has a high tensile strength, but it's very slow growing, so it's going to be outcompeted, right? Turkey tail is very high. Reishi mushrooms are very high. But tiger sawgill is extremely high. I think we tested it at Clemson. It was 800,000 to 1 million times its own weight, which they said was equivalent to the strand of steel. So we are not one mycelium, like particles bound together. And that's what makes it so good, because it can also eat, or adhere to hardwoods, conifers, a whole different range of substrates, which is unheard of. And then its ability to be very fast, high tensile strength. I mean, I'm using that fungus not only for microremediation, but I'm also using it for building materials. So I've got a couple projects going on with one or two companies, and also Clemson University, the engineering department. We're looking at different building materials. which is insane. So this mushroom was just a, you know, it had a big S on its chest. You know, it was out there, and we just realized that, you know, what it can do. But we had to put it through the gym. You know, we had to test it out and see what it could do.
Yeah, I got to tell you, there's no doubt that it's fantastic to hear that there are organizations like I know IKEA made an announcement a while back that all of their packaging material is going to be mushroom based, and so it's beautiful to hear that taking styrofoam that had been shipped arguably all over the world at an enormous amount be replaced with a product material that's made out of mushrooms. So building material out of it is delightful to hear, instead of having some sort of toxic insulation in your walls. Your walls are built out of insulated bricks out of mushrooms. How fantastic. How far along are we on that?
Well, I made some clay bricks with the National Brick Research Center with tiger sawgill and clay, but I haven't made them with, you know, solely like mushroom bricks. Somebody just put in there insulation for a cob house, mycelium and substrate sheets. Exactly. That's something Clumps and I are working on this semester. We've got a whole bunch of students working on things like that, like insulation, you know, some different brick structures, you know, and the bricks that we made just out of sawdust and fungus almost broke their bandsaw in the engineering department. That's how strong they were. And they were so happy to tell me. And so was I. I was like beaming like a proud father, you know, like, yeah, my kid almost, you know, broke your bandsaw, you know. Oh, and costs, yeah, they're biodegradable, Kimberly. But I recently did an interview for Wired. Was it Wired? Is that a name of a magazine? Wired? Yeah, I think so. Yeah, they interviewed me about the mushroom coffins thing. So you're gonna have to wait to see what I said.
When's that supposed to be coming out in Wired?
I don't know. I'll text him, and he interviewed me like 3 times. So it was essentially about the claims that the company was making on the website. And I'm a pretty honest guy. So maybe that'll tell you, maybe that'll give a little bit of a hint about it. Now, hey, listen, I'd be buried in a mushroom coffin. It's biodegradable, but I think the claims were a little lofty on the website. So that's what they were asking about, is the claims, not about the fact that it was a cool idea. I think it was a great idea. So I'm sure when it comes out, I'm going to get some phone calls from that company.
Well, you always start with the, in my humble opinion. And, you're good to go.
As far, yeah, I don't know what the Congress answer is as far as my knowledge, or I'm not aware. There's some go-to answers there.
Yep. Well, I can appreciate, wanting to chat with you multiple times, because there's so much information that you went over tonight, that trying to keep up with you was a fun ride in all of the different, not only the amazing, pictures in terms of their colors and just artistically how gorgeous nature is, but how complicated it all is, and the interaction between the viruses and the bacteria and all of it. So it really is fantastic. You had mentioned, and raise your hand if you need to run. We'll gladly let you go, but we've got a few more questions here. So if you've got a little bit more time, and folks, if you want to stick around with us.
No, I'm good. Yeah, I'm good.
All right, great. So you had mentioned chicken coop and using fungus to inoculate chickens and their space, right? Their chicken coops. So is there a particular one that you were talking about there?
It depends on the slope area. We use King Stropharia in very flat level ground. And again, I would use something like tiger sawgill and something with a slope, right? It just really depends. King Stropharia was better at killing and immobilizing E. coli, but it wasn't as good as site stabilization. So if you have a little bit of a slope, tiger sawgill. So
I got you. There was a question that came up about why morels are so tough to grow.
Yeah, there's actually a chapter on that in my book. That's the shameless self-promotion for that, because it's very complicated. Can I promote another lecture that I'm giving in a couple weeks? I don't know if I can.
Absolutely. I think we'd all like to go to that one too.
That one is called Morel Madness. It's basically me talking about predicting the morel season for this year, but I also talk a little bit about how morels are cultivated near the end. And the chapter in my book is in depth. I couldn't answer it, but what I would try to sum it up is that morels are mycorrhizal potentially at first, meaning they associate with trees, and then they associate with trees that are in decline, which then turns them into a saprophyte or eating dead material. But they also associate or are predatory on the bacteria that are on the roots of the trees. So it's like multiple levels of biology. And there's also ground freeze, there's chill hours involved. They have to have specific chill hours during the winter to fruit properly, and then have those bacterial populations around. There's ways around all this. There's my method and a Chinese method, which is essentially the same thing, just flipped upside down. And so, yeah, it can be done. It's not as efficient or as reliable as, say, shiitakes, but some have pulled it off, and we've got some beds going now at Mushroom Mountain, and I did not supervise the installation this year. I wasn't there, but my team just put some in. So I hope to see if some pop out of there. But I would say with a little bit more, research and people playing around with the information that I provided, because that's the only way it will fruit. That's been proven in a laboratory of basically how it has to grow a certain way. That if you put together the right combination of players on the field, that it can happen very reliably. That's what I'm hoping for.
Nice. Well, we're looking forward to hearing your results as those beds produce for you. There was a question about fairy circles. If there are any particular mushrooms, any particular fungus that produce those versus others.
Yeah, actually, the most commonly consumed poisonous mushroom in the southeast is a green spored parasol, and I'll put that one in here. You can type this one. So, fairy ring. Search that in the chat box. You're gonna see it. You're gonna see what it is. It has green spores. It's got a green spore print, and it'll make you sick. It's one of 2 green spored mushrooms in the U.S. You can't go wrong here from steering clear of this one, right? It's a very big mushroom. It makes fairy arcs, like arcs or rings, and they get massive, like the size of a plate. There are a lot of mushrooms can make fairy rings. Agaricus, or little button mushrooms, they can make fairy rings. Bluets, I've seen coral mushrooms. I've seen a lot of different species do it, but that is the most commonly common formed fairy ring, at least in the southeast. And they're huge. They get big and they're kind of white and scaly in the middle. And when they open up and get flat, if you put the gills down on aluminum foil or white paper, it'll turn it like a pale green. Right? Stay away from that one.
Yeah. So that brings up foraging. I know that you've been teaching people how to forage safely for a long time now. And so is there is there a particular certification that is nationally recognized as for foraging, so you do stay away from the green spore prints type mushrooms and other techniques to find out what you should not go anywhere near?
We offer, I tell you, I wouldn't sell from the stage, but I have to because we have the only one. It's not nationally recognized, but the FDA endorsed it because they came and talked, they saw one of my classes. It's a state-by-state approved mushroom identification course, but that's only if you're selling mushrooms, all right? If you're putting mushrooms into commerce, you have to take a course to be considered, quote, an expert that has passed an exam, all right? And there's other people giving courses. I'm not just gonna put mine up there. But ours is the only multi-state course. So we're certified in all these different states. You can also take a class. We offer in-person and Zoom classes on just, hey, I want to learn how to identify 50 mushrooms, including the toxic ones. And after one day, I feel safe going out in the woods and eating mushrooms and feeding my family. You know, we do that too. And mushroom identification is easier than you think, especially for edible and poisonous and deadly. I cover that in one day. But if you want to sell them, that's a 2-day class. And you have to take an exam. That's not easy. But there's a high pass rate, I'll tell you that. Because, you know, I'm very passionate about it, but I'm also, you know, I like to see good grades. So I really make sure everybody in the class, like, really, you know, is following along. They get good notes, and we have a lot of fun during the class. You know, doesn't seem like work when you're having fun.
That's right. That's right. It's wonderful that you've taken a passion of yours and been able to make it into a quite a successful vocation as well. And the research that you're putting together is beautiful to see. Looking forward to seeing the something that we can actually touch and use. You know, kind of like we were talking about the varroa mite and that kind of zombie maker out of pests and.
Well, listen, that spotted lanternfly is headed your way. I've had a couple contacts that have come to me about doing some trials right there in North Carolina to kind of put up the wall there, you know, the biological wall. So we're gonna, there are some muscadine growers. So we're gonna look at, you know, what we can do to protect some initial crops, and then see what we can do about get an emergency use permit from the state. You know, I don't think we need to wait, and because the chemicals are just, it's going to be too late. You know, I put my website up there just because people are asking.
Right. Yeah. If you guys don't know Mushroom Mountain, definitely look up Mushroom Mountain. That's where Trad has all his contact info. And if you're looking for address or anything else over there.
Oh, hold on. I think I had, the things I were posting with, I was posting the whole time and host and panelist. Whoops. Oh, well.
All right. So you were sharing that with me. That I appreciate that.
Damn, I've been typing the whole, guys. Sorry. I'm sorry. I was putting, I've been answering your questions too.
I've been trying to repeat what you've been saying with these answers. So, you know, with cultures, is there a particular? I know that you can get spawn in like sawdust. You can get plugs that look like little dowels, right? Little wooden dowels that folks can drill into logs and then put in there and cover up with wax. Is there a particular you have a preference for one over the other, and where folks get that?
Probably unbiased, but you could probably find them at Mushroom Mountain. There's a company in South Carolina. But plugs, I would say you can get the same species on plugs or sawdust. Okay, they're two different forms. Plugs, I would say, if you just wanted to, every pack of plugs will do about 10 linear feet of logs, no matter how you cut them. That's for if you want to do a couple logs. All right. And I'm going to go into the metrics here because it's important. Let's say a pack of plugs costs you 15 bucks, 10 linear feet. The sawdust, you can do 100 linear feet for 25 bucks. All right. You do a lot more. It's because it's cheaper to make in the lab. It's cheaper for anybody and us to make, and it's easier to kind of inject them into the holes. So it's just a matter of, and you know, the sawdust can last for a year or two in the fridge. If you don't use it all, you know, you just roll it up, put it in the refrigerator and take it out and do it the following year, or you give it to somebody, your neighbor, you know. But economically, it makes more sense to do the bag of sawdust. If you're doing, that's shiitake. If oysters, same thing, but if you buy, let's say, a bag of oyster sawdust, you can do logs, you can do cardboard, you can do paper, you can do straw. You know, you can try all kinds of cool things with a bag of oyster spawn. That's like a bag of gold to me. Because shiitakes are just growing wood, but oyster mushrooms are growing just about anything, right?
What about chaga and lion's manes? I know that I actually just inoculated a bunch of logs with some lion's mane myself, and I'm looking forward to seeing that. I'm allergic to shellfish, so lobster is not on my menu, but I understand that lion's mane has got that lobster flavor, so I'm looking forward to trying that out.
Yeah, lion's mane is really good. We grow it outdoors on hardwoods. It likes to grow on oak and cherry and maple, maybe some beech. Larger chunks that are just kind of sitting in the ground or slightly buried. And I see someone said lion's mane crab cakes. Man, they are good. And I've even seen some people make like a pulled pork, because it kind of has that, you know, if you pull it apart, it's got that texture. So if you cook it and kind of, you know, caramelize it a little bit and make some barbecue sauce, you can make like a pulled pork kind of product. It's a great mushroom. We grow them indoors. It's got nerve growth factor in it, which has been shown to re-stimulate the reproduction of adult brain stem cells. It's just a phenomenal, that is in, when someone says, do you grow magic mushrooms? I'm like, every mushroom is magical, right? They all have a gift.
That is fantastic. I know plenty of them taste great, and I'm looking forward to trying that myself. So crab cakes haven't been on the menu, so lion's mane crab cake, I'm gonna have to look that up. Mycorrhizae, is there an easy way to add that to our gardens and our compost and everything else? Is there like a mycorrhizae tea that we can make?
It would be difficult to make one yourself. I mean, we've got mycorrhizae at the farm online, but I'm actually working on a book, because I'll be honest that I'm not satisfied with you know, the products that we have to buy bulk mycorrhizae in, you know, and it's expensive. So we just sell it in little, like, you know, 1 pound bags, so people can afford it. It goes a long way, but who's going to buy 50 to 150 pounds of it at a time, you know? So we buy it and repackage it. I'm working on my second book, which also discusses how to cultivate your own mycorrhizae at home. And it's a bit of a process. It would be hard to try to, you know, lay it all out in a minute. But if essentially mycorrhizae are formed on root fragments, on the new growth of roots. So you would have to grow out some plants. Annuals are different than, you know, shrubs and trees. There's endomycorrhizae, there's ectomycorrhizae. So you really have to know, like, what are you trying to grow? And, you know, then you would have to harvest the roots of those plants, chop them up, and then put them in with a seed starting mix or the the plant mix for the next generation of plants. Does that make sense? So they would leap off onto the roots of the next generation, but there would have to be some root harvesting involved. So it could be a challenge to create your own mycorrhizae home for now, but I'm working on a whole section of for my new book on how to do that. I've got some steps that would help.
Fantastic. We're looking forward to seeing that. There's no question.
I love that. That chapter is going to be cool. I can't wait.
I tell you, we can keep going, but I'd like to thank you right now and thank everybody for sticking with us for this presentation. I know we were all looking forward to hearing you present, Chad. You started talking about Morel Madness. Where are we going to be able to see that?
I'll put the website in there. You could go there. I know you're going to be a little bit apprehensive about putting in, like, what's going to pop up when I put scumsonline.com.
Is that what it is?
Yeah, it's South Carolina Upstate Mycological Society online. It's a clever name. That's based out of Clemson, and Julia and I are really good friends, and she's on there, and she'll be administrating. But I'll talk. I've got some really cool stuff on morels. I mean, if you like morels, you don't know what they are, like curious, and maybe want to go out and find some, you got to come to that, because I give all the secrets. I give everything at that lecture. It's just an incredible series of images and data and things that I've collected over the years.
Very cool. We're looking forward to it. So it's scumsonline.com.
Yeah, that's it.
All right. So they're a .com. They're not a .org.
Yeah, .com.
Okay. So
why don't we do one or two final questions, and then we can wrap it up. Is that okay?
Yeah, absolutely. The more the better. Thank you for that.
And anybody can reach me through, at Mushroom Mountain. There's forms, online forms there. We have a big department there that kind of handles different kinds of questions, so.
Yeah, there was George had a question about the orange mold that you showed earlier, looking similar to reddening of a regular parasol mushroom. He's asking regarding one of his substrate bags that he's seen.
An orange mold? I showed an orange mold?
That it kind of hardened a mushroom. Wasn't it like a combination of
Hypomyces? Yeah, that's a mycoparasite. So it
oh man, so you froze up there for a moment right when you were answering that question. So if you said it was a mycoparasite, that's where we lost you for a second.
Yeah. So that's a mycoparasite. So that particular fungus only infects a particular genus of mushrooms. So it won't grow on any.
Okay, so that was not on a parasol mushroom?
No, that was on a Russula. I'll put that in there. Russula.
Yeah, so don't put it in the chat, put it in the Q&A. Yeah, folks can't see the chat, only we can see the chat.
Well, shit. I mean, shoot.
That's why I wanted to spell out the scumsonline.com, make sure people can see where presentations can be.
I put it into the setting to everyone.
Okay.
Everyone should be able to see that.
Oh, okay. Yeah, I see you having it on everyone. Good job. Well, folks, I appreciate you guys coming out and seeing with us today.
Going down. Should I leave tree stumps in my yard?
Yeah, it was actually a good question. He started asking about a tree stump, and then halfway through the presentation, he realized he's just going to leave his tree stump. He's not going to remove it. Let all the mycelium do their job.
Yeah, best surface to do is sport print on. I like aluminum foil. Let it dry, and then you can put it in a Ziploc bag in the fridge. How do I get spores? Daniel? I hate saying it.
You know, I got to tell you guys, I've been down to Mushroom Mountain. It's a fabulous spot that that Trad has, and they've got obviously a lot of the spores, both in the mycelium in the spore bags, as well as in little plugs. But it obviously makes a whole lot more sense to get it. It goes a whole lot more. Talk about having a mushroom log inoculating party. Those bags will go a long way with a lot of logs. Just keep in mind, you do need fresh, new logs or fresh chips if you're inoculating stuff, because of course every other fungus is trying to inoculate all that stuff too when it is out there trying to reproduce. So as soon as you get, let's say, a log, a tree cut, a big branch cut, whatever it is, you only have, what do you have, about 4, 5, 6 weeks? How long do you think you have, Trad, before it really isn't worthwhile trying to inoculate?
It depends on the hardness of the hardwood. You know, if it's like poplar or something soft, sweet gum, you probably have 4 weeks in the winter. If it's something like an oak, I mean, you might have up to 2 months, maybe 3 months, you know, in the winter. Now, things accelerate in the summer. So you have to, maybe sweet gum, you want to do it in 2 to 3 weeks, and oak within a month. Oh, I have a copy of my, can I put a copy? I should have put this up earlier, but I'll put that up there. Because there was a lot of questions about cultivation. And honestly, if you want to cultivate, just my book's a fairly good resource there to get started.
Yeah, so folks, it's definitely the quicker you can to get in there, the better on if you're going to inoculate logs. It's definitely a competition world out there with others trying to make it in there. What a fantastic, you know, all right, you cultured that directly. That wasn't just how it showed up on an auger.
Yeah, I cut that out. This is what I do on a Friday night, man.
Nice. Well, we're glad that you do that, man. We are very glad. And keep us posted. We'd love to have you back sometime to let us know about all the research that you are doing and updates on it. So for all of you guys that don't follow him online, you definitely should look for Trad. He's easy to find on social media, as well as their website and the company at Mushroom Mountain. Hey, Trad, thanks again. We really appreciate you coming out. Everybody else, thanks. And for the folks who are local, we'd love to have you come and join us, not only in these presentations, but also in our efforts to bring presentations like this to you. So thanks so much, everybody. You guys all have a wonderful evening. Trad, take care.
Thanks for having me tonight, too. And hey, if you get the link to the video, share it with other people that couldn't make it. Yeah.
Absolutely. It's going to be on our website. So hawkncwf.org is our website. And you can find it also on Facebook. If you look Habitat and Wildlife Keepers in Matthews, that is us. And so our website's got all of our presentations, including this one. Hopefully that it worked, it recorded, and we'll go ahead and post it. Hey, thanks again, guys. Y'all have a wonderful evening. Appreciate it.
All right. Good
Bye-bye.
night.